we will actually describe here today about the cell envelope. Cell envelope is the entire layer that actually cover the cytoplasm. So within the cell envelope, you have several layers. We're going to go through one by one. Now, first of all, this is, I think everyone familiar with that. We just simply go through it. This is the bacterial cells. Bacterial cells has many organelles. And you will see first thing the flagella, very long. Actually, it's beeping that you can allow it to move forward. You have the so-called pillars or pili, whatever. The pillars is something that important for conjugations to transmit the genetic materials. And we will come with that next week. And you will have the chromosome. Sometimes you describe this as chromosome, or that is these particular regions that contain the chromosome, we call the nucleoid, right? With that, we also have a lot of ribosome. The dot normally indicates a ribosome, and also have some other like plasmic inclusion body that present in the cytoplasm. Other than that, you will have this membrane which is a filament extending out from the cell's wall, and this is also important for which is slightly pathogenic, and also we will come with this next week. Now, today's we only actually looking at the membrane, the cell envelope. The cell envelope contain here plasma membrane, cell wall, capsule, also part of the cell envelope, but we will have this one next week as well. Alright, so this is the EM. Some EM which has a very good resolutions, you can see a layer of the cell membrane that present in the envelope. So now you've got some idea where is the envelope positions. First thing, we go from inner to outer. The inner, close next to the cytoplasm, is called cytoplasmic membrane. So human also have cell membrane. Normally for human or eukaryote, we call cell membrane or plasma membrane. Sometimes if we use it loosely, we also can call this cytoplasmic membrane. From a bacterial, call it plasma membrane, cell membrane. But I would rather call it cytoplasmic membrane or cell membrane. I prefer to use plasma membrane to describe the eukaryote cells. Okay, so we call cytoplasmic membrane. Let's say that if during the exam, if you use plasma membrane, better not, but I will not deduct your mark if I know what you're referring to. So this is the boundary, it's called that isolate the cytoplasm from the external environment. So this is the inner membrane that actually just next to the cytoplasm, which is here. So this membrane, very similar to the eukaryote cell membrane, is formed by the phospholipid bilayer. So it's the one layer, second layer, is a phospholipid lined up, very organized in a way. And within this phospholipid bilayer, that has a lot of protein, a modified protein that has been inserted in the membrane. Some of them is lying on the surface, and some of them actually inserted into the lipid bilayer. And all of them, they will have their own functions. With this kind of protein, you can have glycoprotein, meaning that a sugar or carbohydrate conjugating with a protein. You will have lipoprotein. Do you have lipoprotein here? Or lipoprotein, which is the lipid plus the protein. No, right? But you have the glycolipid, meaning that the lipid here actually with the sugar. So this is called glycolipid. And there's a lot of this. And some of them which is very important because they form, this kind of protein form a channel, and this channel actually used for the transportations. Mainly, first thing is the nutrient uptake for the bacterial cells. Right, and therefore they are selectively whether permeable to sugar and protein, while that allow the gas and water freely pass through. Why? Because gas and water, they are small molecule plus they have no charge. In order to pass through the lipid bilayer, you require something that is at least is hydrophobic when you're passing through at the center. Therefore, like gases without any charge, they can easily pass through the lipid bilayer. So in this lipid bilayer, they actually call it amphibatic. Amphibatic meaning that it's half hydrophobic and half hydrophilic. Okay, half and half. Why? Because the one that are facing this phospholipid, the head and tail, normally we describe it head and tail. The head part is hydrophilic, which is facing the outer environment and also facing the cytoplasm, which is facing a lot of the water-soluble environment. But in the inner of the phospholipid, they have the fatty acid tail, and this is highly hydrophobic. Alright, so therefore they have hydrophilic and hydrophobic, and it's called amphibatic. So is the plasma membrane, what is that? I think I describe it as a functions to make it as a barrier, not allow every molecule passing through, selectively selecting the molecule to pass through the membrane, because you cannot allow everything to pass through, especially water. If you allow the water passing through easily, and the cell will burst. Secondly, and what is this saying here? Okay, it can be used during the cell divisions because two daughter cells, normally they will have partitions to separate two daughter cells. The partition is formed by the lipid bilayer. So also important for the cell divisions. Now, talking about the phospholipid. The phospholipid here, and one thing, the characteristic of bacterial phospholipid or the bacterial cell membrane, cytoplasmic membrane, it doesn't have this cholesterol. We have the cholesterol in our cell membrane. For all eukaryote, they have the cholesterol in the cell membrane. The cholesterol serve a very important purpose. What is the purpose they serve? They actually provide the membrane fluidity, right? So at high or low temperature, cholesterol are important in high or low temperature. For example, in high temperature, the cholesterol actually increase the adhesion, meaning that it connecting the phospholipid together, not because it's during the high temperature, all the phospholipid try to separate it from each other and they become very active, moving around. But cholesterol help to adhere from each other so that it stay as a structure of a cell membrane. At low temperature, because at low temperature, it's become frozen and it's become very rigid, easy to be damaged. What cholesterol do in eukaryote actually is make it fluid and allow also not so rigid. It provide the fluidity to the cell membrane. And because when you are rigid, meaning that you have all the phospholipid lying up like this. So it's been organized in one way and easy to be broken down. But if you have the cholesterol inserted in between, so you are making it a different actually orientations as well as the organizations and make it more fluid and during the low temperature. Now, thinking about the bacteria, so important that the eukaryote cells having the cholesterol, what happened to the bacteria without the cholesterol? And it has, of course, the phospholipid. The phospholipid of the bacteria will have this structure. Normally the circular is a polar head group. And then you will have the phosphide group. And you have a glycerol. The glycerol connect to the two fatty acid tail. And the connection is through the ester linkage. All right, as simple as that. And this phospholipid of the bacterials, it's the same, exactly the same as the one that you see in eukaryote. Now, we're talking about what happens if the bacteria do not have the cholesterol, but they have something like structurally similar like the cholesterol. We call it the hopanoid. A lot of, not all, quite a number of bacterials, they steroid cholesterol belong to steroid. They cannot synthesize steroid, right? But actually they can derive it from their environment. They can take in. Cyanobacterials, they do not produce the cholesterol, but they produce something called hopanoid. Hopanoid is something that's structurally similar like cholesterol. If you look at the cholesterol here, this is cholesterol, sorry. This is hopanoid, which is contain 1, 2, 3, 4 cyclohexane. So this is cyclohexane ring and one cyclopentane ring. And you look at it, the ring has got 1, 2, 3, 4 of the hexane ring and one pentane. So structurally, they are quite similar. So instead of the cholesterol, some bacteria, they use the hopanoid. And we believe that they will serve to the similar function like cholesterol to actually provide the fluidity to the cell membrane and also to actually connecting between the lipid to have enhanced the adhesions of the phospholipid and limit the penetration, of course. We are already told that the cell membrane actually is formed as a barrier, not allow all the molecule to pass through. All right. Now, we talk about the protein in the cell membrane. When we talk about the protein, the only protein, of course, proteins serve different functions. They can be enzyme, sometimes. They can be receptor, but here we are talking something more important. They are the transporter on the cell membrane. When we talk about the transporter and the way of the transporter, how do they do? We can divide them into at least 3 kind of like transportation of the molecule into the bacterial cells. The first one is the passive diffusions. You learned about that, right? Passive diffusions, osmosis. So when you have heard passive, meaning that they do not need any external force to bring in the molecule. Let's say bring in and out of the molecule into the cells and out of the cells. So it's passive, doesn't require energy. So it's called passive. So it's diffusion, meaning that the molecule moving from high concentration to low concentration along the concentration gradient. That's why it's diffusion. So some of the molecules such as water, ethanol, gases, as I mentioned earlier, gases do not have any charge. Same as the water, small, ethanol also, small and no charge. They can easily diffuse in and out without any protein that required. And also they are moving from high to low concentrations. Okay, facilitated diffusion. The diffusion requires something to facilitate it. The protein and protein channels has been used to facilitate the transportation of the molecule into or out of the cells. Now, why they require protein carrier or protein channel as the one that's showing here? And because these kind of molecules are big and they are polar, meaning they have charge, and they cannot easily diffuse into the cells. Therefore, they require the protein to carry them into the cells. Normally, this is the protein channel will have a pole at the center that allow the molecule to pass through. Normally, this protein will change when they're receiving the whatever the molecule, let's say it's a nutrient, they will actually, there is a pouch, something to accept, and they have a conformation change, and then it will actually go through and release that into the cytoplasm. Those are really big or polar molecules such as glucose, amino acid, sodiums, ions, and chloride. right? All those are slightly bigger, and also they are charged. They're not easily to diffuse through the membrane. Now, I think these two no problem. The third one is active transport. Active transport that different from passive. As I mentioned, passive, no external force required, right? But active transport require something to make it as a push. That means it requires energy to transport the molecule. The energy here is the ATP. ATP hydrolysis. And this is the difference between active and passive, require energy. The second difference is that it also has a concentration gradient, but the concentration gradient run oppositely, meaning that the molecule transport from low concentration to high concentration against the concentrations gradient. So these are the two that are different from passive diffusions. And of course, they also require a protein channel. The example here is the ABC transporter that can actually transport some molecule here, which indicate amino acid, peptide, lipid, bacteriocin. Bacteriocin is used to kill the bacteria, organic anions, which is like cyclin AMP or cyclin GMP. Right, so there are two different, for passive transport and active transport, both of them have the protein channel that require, but the difference between these two is that active transport run against the gradient concentration, and it requires the ATP hydrolysis, meaning the energy to allow the protein for the transportations. Because the reason is running from bringing in from low concentration to high concentration, it requires energy. As you can see, for active transport, there are three types: primary active transport, secondary active transport, and the last one is group translocations. For primary active transport, and very similar, it just requires a protein channel, which here is a protein pump. It pumping this molecule, which is a proton, the hydrogen ion, from low to high concentration against the concentration gradient, and it requires the energy, the field is the energy. All right. Now, secondary transport, active transport, what is the difference? Here you have a co-transporter. So this proton pump is similar like that. Similar like this primary active transport. The proton is pumping from inside to out, which has lower concentration inside, higher concentration outside. Therefore, you require the energy to pump it out. When you're pumping this proton out, right, you create that is a high proton concentration outside. Now, not too far from this proton pump, there is a co-transporter. And here, the co-transporter is called lactose permease. This lactose permease, as what is named, is transporting the lactose into the cells. It's sort of like uptake of the nutrients. Now, how do they do that? Because this proton accumulate, because keep pumping out, using the energy pumping out, it create high concentrations. So high concentrations will automatically tend to flow back to low concentration if there is a transporter that allow it to do so. So this lactose permease transporting the proton from high to low concentration. At the same time, it brings in the lactose together. So with that, is called secondary active transport, allow them to actually accompany with two transporter, and you allowed to bring in the lactose. All right, and why we still call it active? Because although this particular co-transporter may not require energy, however, you have a proton pump actually as a prerequisite to allow him to pump the proton out, so that the proton high concentration here come back and bringing the lactose come in. Okay, and the last called group translocation for the active transport. This one slightly different from the primary active transport. is because they modify the molecule. The molecule here going to be transported into the cells is glucose. All right, they actually very typical. All the textbook that you saw, they are giving you this particular example called PEP enzyme. What does PEP stand for? Phosphoenolpyruvate. And this is part of the sugar phosphortransferase system. What does it mean is that this particular enzyme is able to transfer a phosphide group, because it's an enzyme, transfer a phosphide group to the glucose. So it is also present as a transporter. So when the glucose try to come in, and it itself has a phosphide group, and the phosphide group will actually breaking into a phosphide group, and this phosphide group, when you breaking into a phosphide group, is generate a lot of energy. So this phosphide group will attach to the glucose during here. So this is the phosphide group, this is the glucose. Inside this PEP, they actually given the phosphide group to the glucose, and therefore outside it is glucose. When they passing through this PEP, come into the cytoplasm, they become the glucose 6-phosphate. So actually going this through, they actually changing and modifying the glucose, the molecule, into a different molecule. And when actually they become the glucose 6-phosphate, there is no way back, they can be going out again. Right? So this is called group translocations. Oops. You're still okay? We still have 10 more slides, roughly 9 slides to go. Very quickly, don't worry. This is something, cell wall, everyone familiar, peptidoglycan. The bacterial cell wall, we call it, is made up with the peptidoglycan, or we sometimes call it murine. And the peptidoglycan, if you look at the structure of peptidoglycan, sorry, slightly tired, it's down. You will have a very basic component called N-acetylglucosamine, NAG, or N-acetylmuronic acid, NAM. This is a very basic unit for the peptidoglycan. In addition of that, this is amino sugar. You will have the peptide, which is the amino acid. Okay, but they form as a short peptide. And then you have the pentaglycine, which is also forming a bridge, and also present in positive bacteria whatsoever. And now, if you're looking at here, I actually divide this one. This is the gram-positive peptidoglycan. This is a gram-negative peptidoglycan. They are slightly different. What are the different? First of all, they are exactly the same in the way of the NAM and NAG. The orientation is one NAM, one NAG, and NAG like this. Now, you're making this, right, the sugar chain, amino sugar chain, but they are in a single chain. There is no network forming to the next of the, what is that? The polymer, they call it polymer. The next polymer. So they actually parallelly align in this way. But you need to actually forming as a network, a peptidoglycan, you need something to connect, right? These two parallel polymer. So this, first of all, beta-1,4 glycosidic bond is this one that is joining this NAM and NAG together. And this polymer joining with another polymer, actually is in the positive, gram-positive, is with the amino acid, or that is a peptide. Now, looking at this, all the NAM, they actually link to a short peptide. These short peptides are forming, there are 4, L-aniline, D-glutamine, L-lysine, D-aniline, like this. The next one, they have the same. The next one also have the same. Then you have the third amino acid L-lysine joining with the D-aniline of the next one. And what are joining them? There are 1, 2, 3, 4, 5 glycine, which is also amino acid. 5 of them actually form the bridge and joining to next one. That's why from this polymer to this polymer, they can be connected by the peptide glycine. As simple as that. And you look at the gram-negative, basically NAN, MAG are exactly the same. And the only difference is the amino acid here. You have the L-aniline, D-glutamine, and this one is called L-diamino pimeric acid, DAP. That's the third one, is the only one that are different from the gram-positive. And also the cross-link is different. They are not using the glycine for the cross-link. In fact, they have the direct cross-link with between the peptide. All right? So these are only differences. Phew. Anyone aware of this? This is a gram stain. I think some of you, not only the first time, second, third time, you have done in your high school, in your pre-U. However, gram stain is very important. So, gram positive, after the gram stain, will show you purple color. Gram negative, will show you pink color, right? E. coli, gram positive or gram negative. You must have used E. coli and have done the gram stain. Is gram positive or gram negative? Some of you say something. Don't worry, you will have using the E. coli in your practical class and do the gram stain. And by then, you will know whether it's E. coli, gram positive or gram negative. Why that gram stain able to distinguish between gram positive and gram negative? We have this name gram positive and gram negative is all because of the gram stain. Because they stain in different color, therefore we can call positive and negative. I think if you're going through that, there are two very important dye. What is the crystal violet and the other is saponin, right? Crystal violet, here, do I need to explain it? I think I need very, very fast. So crystal violet is the first dye that you already have the bacterial cells. Maybe you doesn't know that whether it's gram positive or gram negative. You just want to see whether it's positive or negative. So you apply the crystal violet. It's just that short dry staining for 1 minute, right? So what happens is that crystal violet will go into the cells and it will stain the whole cells, become purple color. Now, you will have the iodine. Iodine is something that makes the staining firm. So you apply it. Iodine is a brown color, you say orange color, but brown color once again, just to make it stick, very, very stick to the purple color stick to the cells. And then you rinse it, and every single, actually in between this staining, you will have the rinsing step with the tap water, and then you will have applied the ethanol and to actually wash out the color. All right. Now, if you're looking at that, some of the bacterial cells still remain as a purple color, that it doesn't wash away the purple color, but some of them become transparent. And lastly, when you apply the saponin, which is in pink color, it's a pink dye, and when you apply 45 seconds, all right, those are transparent, will again being stained in the pink color. Those in already purple color, or those it can stain with the saponin, but because of the purple color is so strong, so it's remain as a purple color. Now, what is the principle behind that? Why important is here, when you rinse with the ethanol, some of this crystal violet can be washed away, right? So why it can be washed away is rely on the thickness of cell wall. And it is the gram positive, right? So it's retained as a crystal violet color, the purple color. meaning that those crystal violet cannot be easily washed away by the ethanol. And because of this, the thickness of the peptidoglycan, so it's not easy to penetrate, like rings and penetrate out of the cells. And however, if you're looking at the gram-negative cells, the thickness of the peptidoglycan is not thick, it's very thin. And it can, when you apply the ethanol, it can be washed away. And therefore, when you apply the softening, and then it will be staining again. Now, these conditions, 60 seconds, 60 seconds, 5 to 10 seconds, or 45 seconds, is it important? Yes or no? Yeah. What is my questions? Are you asking? I mean the second, the procedure, the step. If in your menu, I say that please use ethanol to apply ethanol onto the cells, 45 seconds. Is it 45 seconds? Sorry, no, 10 seconds. Let's say if you apply the ethanol in this step, rinsing it, and then to 1 minute, what happened? I hear many of you are saying something, but I do not know the answer. You will wash off the purple color, good. It will wash off the purple color. So therefore, every single, when you stated it here is very crucial. Sometimes I say that, oh, I have a gram, sort of like I have a gram positive, but how come my gram positive cell is pink color? Because when you're applying, because there is a thickness of the peptidoglycan, will sort of like using 10 seconds to wash off, it's not easily to wash off the crystal violet. However, if you apply for 1 minute or over using the ethanol to wash these gram-positive cells, I think no matter what, all the crystal violet will be gone. All right? So it depends on how you actually control every single step. If you do every single step properly, you will have the expected result. Otherwise, you will have the gram-positive become pink. Or even that, I do not know what kind of expected. Sometimes this can be purple as well. Sometimes students get it. All right, so next one. Teichoic acid. Teichoic acid is one of the components that present in the cell envelope. Where does this present? Is actually present in the gram-positive. My last slide. Or do not have that. I deleted that. So this table can see that comparing gram-positive and gram-negative bacterial, because the peptidoglycan is very thick, so the cell envelope has the dry weight for the cells. The envelope is actually filling up from 50 to 90% for the entire cells. For gram-negative, because the peptidoglycan is very thin layer, so it only has 10% from the total cells weight. And the number of peptidoglycan layer, this is more than 50 layer or close to 50 layers. This is 1 or 2 layer. See, the thickness are very different. However, and also that we talk about this teichoic acid, there are something that present in gram-positive but not in gram-negative. So what are the teichoic acid? It is a linear polyoid phosphide polymer. It's sort of like polymer. It's negatively charged, and it's important. There are 2 types. One is called lipoteichoic acid. The other one is called wall teichoic acid. As you can see from, I think better from this diagram. And this diagram, you have the lipoteichoic acid, right? This lipoteichoic acid actually is long and is all the way penetrating, passing through the cell wall, the peptidoglycan, and actually anchor at the cell, the inner membrane, not inner, the cell membrane. So this is lipoteichoic acid. However, you also have the very short one, we call that wall teichoic acid, and it is directly actually conjugating with the peptidoglycan, which is pointed that correctly, right? Yeah, this one, wall-tackle acid. So there are two types. One is directly linked and bind to this membrane. The other one is directly bind to the peptidoglycan. So people think that it has maintained the cell shape, is regulate during the cell divisions, and also because they are protruding on the surface of the gram-positive bacteria, it may actually can make contact to the whole cells tissue. Now, we have more fun. We talk about the periplasmic space. Where is periplasmic space? Periplasmic space is between, at the center of the envelope. Both gram-positive, gram-positive, negative bacteria has this periplasmic space. But it is more apparent that in the gram-negative bacteria, they have a very obvious periplasmic. This is a periplasmic space, like between the outer and inner membrane. For a while, they thought there is no periplasmic space in the gram-positive cell wall. However, later they found there is a periplasmic space between the peptidoglycan and also the cell membrane. But it's just that here a very thin, very thin. It's not easily can be seen even in the electron microgram. But there is the existence of this periplasmic space in gram-positive cell. Now, what is this? Why there is a space in between? How important of this periplasm or periplasmic space? And if you think about the uptake of the nutrient, you see the nutrient coming from pollen. Pollen maybe take up the nutrient, go, but they need to actually be transported into the cytoplasm, right? Otherwise, the cell do not have the nutrient to generate energy. But is the nutrients coming here? There is a large space here. And what they do here, maybe they're floating around here. They'll never be able to pick up and go into the cell membrane. But in actual fact, nutrient uptake by the protein channel, and it's also been very actually facilitated by the periplasmic the network, and it can go actually transporting it down all the way to the inner membrane and to the cytoplasm. So it's very much is being monitored and facilitated by the periplasmic. And in here, this is not just an empty space. There is a lot of protein. There is a measures of the could be some carbohydrate or protein present a lot of things present here. Now, we want to know how important it is. 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5. One by one. Nutrient digestions. There is a lot of very comprehensive, but I think you can actually refer whatever happened here to whatever's written here. There is an enzyme which can use to digest the nutrient when they're being picked up. So when they're being digested, they can become smaller and easily pass through the inner membrane. Enzyme also called detoxifications. Beta-lactamase can digest penicillin. We know that penicillin is antibiotic. We apply to the bacteria, but some bacteria has beta-lactamase. That is an enzyme to degrade the penicillin. What happens is that this is the antibiotic. Let's say this is a penicillin. It's picked up by pollen. It's go in, and you will facing a so-called penicillin binding protein. This protein is usually used to actually make the peptidoglycan, required for making the peptidoglycan. When the penicillin bind on this PBP, and this PBB actually inhibiting its functions and is no longer be able to use for the making the peptidoglycan. So what happens is that then if you cannot form a proper cell wall, a peptidoglycan, and it will collapse. So penicillin is used to actually destroy the cell wall, which is good because human do not have cell wall, so it's only targeting the bacterials. And it doesn't actually effective when the cells are not, the bacterial cells are not growing. So when the bacterial cells are growing, the cell wall is also growing and is required this penicillin binding protein. So it's only penicillin effective when the cell wall are growing, the cells are dividing. Then it can stop the cell dividing first of all. All right, so it contains this periplasmic space, contain the beta-lactamase, and very important because all this antibiotic not necessary to go all the way to cytoplasm, because some of the antibiotic actually to stop their ribosome function. So they are not allowed them to come to the stage that they go into the cytoplasm. Actually, they degrading the antibiotic at the periplasmic space. It's facilitated transfer. As I say that there are made some protein carrier that actually can transfer, bind to this pollen and then be able to take the nutrient and then to the inner membrane. So they actually has a sugar, iron, amino acid can be transferred to the periplasmic space. And also it's become the bacterial toxin can be stored. They can be produced, a lot of bacterial toxins and stored in this periplasmic space. When it been required, and it will immediately release out of the toxin. All right. And finally, chemoreceptor, which is the environmental sensor. What happened is has called RCS system. We go for the next slide. Briefly describe what is RCS. This is a very simple diagram. In actual fact, it's more complicated than this. I only talk about two proteins here. One is the RCS, F. RCS stands for regulator of capsule system. This RCF system is used to sense of any stress to the outer membrane. Let's say, for example, if that is normal, there is no stress, for example, stress from anything that attack, right, to breaking out the, for example, you apply the, a lot of like ethanol. So ethanol actually applying to the bacterial membrane, it actually can collapse the membrane. Such, if that happen, that is called stress. So RCF is what to do. This RCF regulatory capsule system that can sense, what happens is that it can change its structure and it contact to the RCF signaling. This RCF signaling is present in the inner membrane. When it's been triggered, what happens is that it can have a formation of capsule. It can form the biofilm. Biofilm is something that can resist the external stress. And it can actually produce flagella to run away and also can produce the toxins. So this is actually is a defense system from the bacterials. And if you have actually, there is a broken of this outer membrane, right? And you will actually change the RCSF, and which is trigger the RCSF signaling start to changing the gene expressions of a lot of bacterials, protein that involved in making the capsule, making the biofilm, making the flagella, and also making the toxin. So this is called a sensor. It's a sensing system for the environment, especially the stress. And this one I'm actually mentioned this because they are present in the periplasmic regions. This is a peptidoglycan, but this is the periplasmic regions. So they have the sensing system in the periplasmic region. All right. Few more slides to go. Very last one, I think two more. One is the LPS, which is extremely important. The other one is lipoprotein, I think. LPS, what does LPS stand for? Lipopolysaccharide. Okay, LPS stands for lipopolysaccharide. It has three components, three parts. And I think the detail of the structure will be mentioned again in next lecture. But first of all, we look at the entire what is LPS. Huge. This is LPS. It's present In the gram-negative bacteria, remember, gram-negative bacteria is because some of the gram-negative bacteria is virulent, we call it, and because they have a lot of this LPS. So why LPS is become very virulent for human? Because our human immune system, especially the innate immune system, can recognize LPS. If our immune system do not recognize LPS, do not react with it, then it is not virulence at all. No harm to us. However, unfortunately, we have some receptor recognize the LPS from the bacteria, and therefore it's become virulence to human. Let's see. And as I mentioned, they have different components. Here I mentioned only two. One is the O antigens present here. It's a saccharide, polysaccharide. It's all sugar unit. And you also have the lipid A. Lipid A, you have the lipid component here, and also which is actually embedded inside the outer membrane. Alright, so this is the entire thing hanging at the outer membrane. On the surface of the bacteria, we call LPS, lipopolysaccharide. And sometimes we also call it another name. What is another name of that? Endotoxin. Endotoxin. Why is it called endotoxin? Because it is toxin to human. That's why I call endotoxin. Why it called endo? Because this is part of because this LPS is part of the structure of the outer membrane bacterial. So it's part of the bacterial structure. So it's called endotoxin. You have another name called exotoxin. Exotoxin is not referring to as like LPS, which is part of the structure of bacteria. Exotoxin is the toxin that purposely being produced by the bacteria and release it out as a toxin, and also cause harm to the human cells. So you have exotoxin, endotoxin. It's called endotoxin because it's part of the structure of the outer membrane. So normally you cannot, some of the gram-negative, you heat it up high as 100 degrees Celsius, but sometimes the LPS is still not be easily destroyed, and it's also create fever in human when we ingest the food contain the LPS. All right. And all right, because I mentioned that why they are virulent to human, because human has the cells such as all these immune cells, monocyte dendritic cells, macrophages. He's hiding macrophages. Is expressing a so-called toll-like receptor. You must have heard about toll-like receptor, because toll-like receptor 4 recognize LPS, especially the lipid A. Okay, so LPS, toll-like receptor 4 recognize LPS, and because of the recognitions of this is a TLR4, toll-like receptor 4. It is present as a dimer. It's supposed to be a monomer when when they are not encountered with the LPS. When they encounter LPS, they form a dimer. So the dimerization here, changing the structure of this, and also eventually changing the structure here. This part, which I think is the C terminals, if I'm not mistaken, actually is inside the cytoplasm. This is a membrane. Membrane somewhere here. This is a receptor which is on the cell membrane, and this part is exposed to the cytoplasm. So actually this part will trigger subsequently the signaling pathway, and the signaling pathway will trigger the pro-inflammatory responses and require, because toll-like receptor 4 tells us there is an invasion of the bacteria because they detect the LPS. So in react to this invasion, we produce a lot of cytokine, which is the inflammatory molecule such as the TNF, tumor necrosis factor alpha, interleukin 1 beta, and interleukin 18. There are some interleukin 1, 6 also part of the cytokine. They actually express during the inflammations, the stage of inflammations. And therefore we have fever, all this. because of a single LPS trickle, the immune cells respond to it dramatically. The next slide is showing how toll-like receptor recognize LPS. Step by step. This is the bacterials, gram-negative bacterials. Of course, when gram-negative bacterials, it could be go into our body. Okay, perhaps we ingest the food into our stomach because of the acid. Maybe the bacteria will purge, whatever, be degrade. And however, this LPS, okay, even that the bacteria destroyed, but the LPS is still there. It's resistant to high temperature, also resistant to acid as well. So this LPS still present in our stomach, and then you go into the bloodstream everywhere, okay? Once it's going to that, in the bloodstream, we also have, of course, immune cells present. You will have a so-called LPB. What is LPB? LPS binding protein, meaning that this LPB recognizes LPS. It found the LPS, is bind on it. All right, and it bind on it, what happened in next? This LPB will bring the LPS to CD14. CD14 is also a cell surface protein on the macrophages, for example, or dendritic cells. So this CD14 recognizes the LPB, which is in white color. It bind on it, and along that, this CD14 on the cell surface, right? And it will actually move, right? The binding moving until they hit the toll-like receptor 4. So when they hit the toll-like receptor 4, and it will bring the LPS to the toll-like receptor 4. With that, you require to recognize the LPS, to actually bind to the LPS. The toll-like receptor has to be bind to the MT2, this protein as a heterodimer. So then they allow the LPS to bind on it. And with this complex, then they will have another one, because this one, they actually repeat again. They will bring more and more to toll-like receptor. So this is repeating to bring more. So you will have, as long as you have two dimer, also have the LPS, they will dimerize like this. So dimerize like this, then will trigger the signaling activations, which I mean the pro-inflammatory response. So this is how it describes what happened here. Okay, the reason is that our immune system are protecting us so well, and our first line defense, our innate immune response, especially macrophage, dendritic cells, and contain the TLR4 on the cell surface, which eventually can recognize all these LPS from degraded, and then these from the bacterials, and then it causes the inflammations in our body. All right, very last one. This is also in the outer membrane of the gram-negative bacterial. It's called the lipoprotein. The lipoprotein is here. So this is the LPS. So the lipoprotein actually is anchored between the outer membrane and also the peptidoglycan. It's actually like holding it and anchor it so that it doesn't actually, the outer membrane will not be fracturated so often. So it actually making the structure firm. So here you show the diagram. It's very clear, this EM showing the outer and inner membrane, and this is the somewhere peptidoglycan is in between these periplasmic regions. So it's just indicate here, right? So this lipoprotein. Lipoprotein has 3 fatty acids that actually holding at the outer membrane, because if this is a lipid bilayer, you need something also lipid, hydrophobic. So they have 3 fatty acids holding here, and also N-terminal that link to the, with the N-terminal link to the fatty acid, and C-terminal bind to the peptidoglycan. So N-terminal here, C-terminal of this lipoprotein bind to the peptidoglycan. So the whole thing is that to maintain the structure, to stabilize the outer membrane by actually holding it with the cell wall. And the last one, which is mentioned here, as I mentioned earlier, you have pollen. Pollen actually can allow the nutrient to come in. It's also allowed the antibiotic to go in. So pollen in a way is a trimeric, meaning that there are 3 protein subunit making this pollen, and it's for any water soluble nutrient can passing through it. You'll find a lot of pollen in the outer membrane. All right. That's all for today. Thank you for staying awake for 2 hours. It's a long hours. Okay then, I'll see you